In the last video, we learned how the first single-celled organisms came into existence, and explored the branches of the evolutionary tree from the ancestor of eukaryotes to the first animals. Now let's find out how these first animals evolved to become the only living member of the genus Homo, Homo sapiens. I know I breezed through the lineage of Eukaryota to Metazoa pretty quickly, but now that we're at the ancestor of all animals, we can slow things down and really examine how these organisms diverged over the next 760 million years. First off, we should figure out what these first animals would have looked like. The common ancestor of all animals was multicellular, but still had a goopy, malleable form like other eukaryotes such as amoebozoa and the ancestor of plants. To know more about exactly what this animal looked like though, we have to look at two of its earliest offshoots, the sponges and the comb jellies. It is highly debated whether Porifera, the sponges, or Ctenophora, the comb jellies, were the first to diverge, but in the phylogenetic tree used in this video, I'm placing the divergence of comb jellies after the divergence of sponges. Whether one split from the first animal before or after the other though, we can infer that the first animal shared certain traits found in both sponges and comb jellies. This is difficult to picture, as sponges and comb jellies are incredibly different from each other. In fact, it's hard to even say if the first animal had nerves like comb jellies, or if it lacked nerves like sponges, and nerves just happened to evolve twice, or that sponges lost their nervous system over time. Now, before I move on, I should mention that the Placozoan Dickinsonia, which is thought to be a sister clade to Eumetazoa and Porifera, is in more recent years looking more likely to be the true first branch of animals, which is interesting because Dickinsonia possesses traits such as a symmetrical body, which wouldn't really be commonly seen for another 200 million years or so. To keep things simple though, I'm going to leave the phylogenetic tree as it is and move on to Eumetazoa. Eumetazoa, or the diploblasts, split from Metazoa 700 million years ago, and branching off from Eumetazoa were Ctenophora, the comb jellies. The sister to Ctenophora, Parahoxozoa, split from the diploblasts 680 million years ago, and then branched into Placozoa, which are very simple animals, and Planulozoa. Planulozoa is a really interesting clade because it is at this point that 560 million years ago, a major branch in animals occurred. On one side is Cnidaria, the phylum that includes coral, jellyfish, and sea anemones. The other side is Bilateria, the clade that includes everything else, and is characterized by the feature of bilateral symmetry, for the most part meaning that these animals had a clear front end and a back end. Bilaterians also had a coelom, a body cavity containing a digestive tract and other organs. Most early bilaterians had a gut that ran all the way through the body, but others had a bag gut with only one opening, meaning its waste came out the same hole its food went in. Other characteristics of the first bilaterian are up for debate, such as whether it was microscopic or macroscopic. We do know the first bilaterian, which we call Urbilaterian, had a mouth and simple eyes near the front of its body, but beyond that it's unknown how complex this animal was. It may have possessed a brain and a central nervous system, a segmented body, a complete digestive tract, and additional sensory appendages, or it may have had a blobby, non-segmented body, a bag gut, and lacked a central nervous system, or it may have been somewhere in the middle. After the appearance of bilaterians, there was another split, separating the phylums Proarticulata and Xenocoelomorpha, which includes achilles and other worms, and the clade Nephrozoa. Nephrozoa is also a major branching point, because it split off into Deuterostomia and Protostomia, the ancestor of mollusks, arthropods, and annelids. The earliest known deuterostome is called Saccharhitis, and it looks like a living nightmare. Anyway, around 540 million years ago, Deuterostomia branched into Chordata and Ambulacraria, which includes Echinodermata, the starfish, sea urchins, sand dollars, and sea cucumbers, and Hemichordata, which contains eggcorn worms. Before we see how Chordata branched into the cephalochordates and the olfactors, let's take a look at their body plan and see how these animals differ from their bilaterian ancestors. The defining features of chordates is that they possess a notochord, a dorsal nerve cord, pharyngeal slits, and a tail. The cephalochordates include an animal called a lancelet, while the olfactors include the tunicates and the first vertebrates. Now, it took a long ways to get here, but after separating ourselves from the likes of amoebas, sponges, jellyfish, and corals, we can talk about the backbone-having animals, the animals that make up the vast majority of all chordates. These first vertebrates had a well-defined head and tail, and even somewhat resembled modern fish at a glance, though they lacked features such as a jaw. That would change though, because 462 million years ago, vertebrata split to include the jawless fish Anatha or the silostomes, hagfish and lampreys, and Gnathostomata, the jawed vertebrates. Here's an interesting one. Branching from Gnathanostomata are the chondrichthyes, or cartilaginous fishes, such as sharks, rays, skates, sawfish, and chimeras. We'll definitely be looking at these animals in more detail sometime soon. We didn't evolve from them though, but instead the Osteichthyes or Eutylostomii, the superclass of fishes that contains more than 90% of vertebrates that are alive today. 420 million years ago, Osteichthyes branched into Sarcopterygi and Actinopterygi. Actinopterygi are the ray-finned fishes, which include animals like tuna, salmon, seahorses, mahi-mahi, and anglerfish. The sister to ray-finned fishes are Sarcopterygi, the lobe-finned fish. Sarcopterygi branched to include Actinistia, the coelacanths, and Rhipidistia, which was formerly called Dipnotetrapodomorpha. Rhipidistria branched to include Tetrapodomorphs and Dipnomorphs, the lungfish. Tetrapodomorpha branched to separate Eotetrapodiforms from extinct lobe-finned fish, including Megalithiidae and Osteolipidae. Then the Eotetrapodiforms branched to separate the Elpistostygalians from extinct lobe-finned fish, including Platycephalichthys. Elpistostygalia then branched to separate Stegocephalia from the extinct lobe-finned fish Panderichthys. Next, Stegocephalia branched to separate tetrapods from extinct lobe-finned fish, including Elgenerpeton, Wachiridae, Colostydae, Ichthyostega, Persiguirhinus, and Buffetidae. Okay, I'm finally done talking about long-dead fish now. That was a lot of fish, but we finally reached the tetrapods, the first four-limbed animals. Of course, the fishes up to this point all had gills, but for members of Tetrapoda to put their limbs to work, they had to be able to breathe air. Early tetrapods still had gills, but they also had an opening called a spiracle, which evolved as a swim bladder and was eventually repurposed as the basis for lungs. As these fish took their first steps in shallow water, they developed neck vertebrae, and other features that would later make them look more like land animals than fish. These early tetrapods, adapted to life in shallow waters, would branch around 350 million years ago to include amniotes, animals adapted to lay eggs on land, and anamniotes, animals adapted to lay eggs in water, which includes amphibians like frogs and salamanders. The land egg layers, amniota, split to include synapsids, and Sauropsida, which includes reptiles like turtles, snakes, crocodiles, dinosaurs, and birds, and an extinct clade called Parareptilia. The synapsids, which split from amniotes 308 to 312 million years ago, are what are known as proto-mammals. They are defined by the temporal fenestra, which is an opening in the skull behind the eyes, and teeth that are differentiated into canines, molars, and incisors. Early synapsids had scaly skin similar to that of a turtle or crocodile, and they laid eggs, but they were warm-blooded and may have had some form of hair. Synapsidia branched to include the Ceciosauria, late Carboniferous and Permian synapsids like Eotheridae and Cassidae, and the Eupelicosaurs. Eupellicosauria includes therapsids and pelycosaurs like Varanopidae and Ophiocodontidae. 275 million years ago, proto-mammals developed limbs oriented beneath the body rather than splayed out like in lizards, in the clade Therapsida. Therapsids include Eutherapsida, Tetraceratops, and Biarmasuchia. Eutherapsida branched to include the Neotherapsids and Dinocephalia. Then Neotherapsida branched to include Anomodonts, which were the dominant terrestrial herbivores at the time, and the Theriodonts. Interestingly, Theriodontia includes Eutheriodontians as well as Gorgonopsians, which were apex predators such as Gorgonops and Sauropterus. Eutheriodontia includes the Cynodonts and Therocephalians, nicknamed the Beastheads, and includes species like Prosterognathus and Moscarhynus kachingi. Cynodontia branched to include Epicynodontia and several extinct therapsids such as Tarasognathus and Trithelodontids. Epicynodontia branched to include Eucynodontians, Gallisaurids, and Thrinaxodontids. And then Eucynodontia branched to include Mammaliaforms, Cynognathians, Trithelodontids, Brazilodontids, and Tritleodontids. So, here we are at the home stretch. No more worms, no more fish, no more mammal-like reptiles. At 220 million years ago, we have reached Mammalia, the class of mammary gland-having animals which branched 160 million years ago to separate the therians from the monotremes, which became platypuses and echidnas. Therians split into eutherians and metatherians, the marsupial relatives, 125 million years ago. Marsupials include animals like kangaroos, possums, bandicoots, and Tasmanian devils. Eutheria, also called placentalia, are placental mammals, and branched to separate Boreoeutherians from Atlantogenata, which includes many animals such as aardvarks, tenrecs, hyraxes, elephants, dugongs, manatees, anteaters, armadillos, and sloths. Boreoeutheria branched 80 to 100 million years ago to separate Euarchontoglires from the Loriciotherians, a huge clade that includes animals like hedgehogs, moles, bats, ungulates, whales, pangolins, bears, cats, and dogs. Euarchontoglires is the superorder that includes lagomorphs, tree shrews, colugos, and primates. It branched into Euarchonta and the Galeoforms, which includes rodents, rabbits, and pikas. Euarchonta includes Scandentians, the tree shrews, and the primatomorphs, which include primates and Dermoptera or colugos. 79 million years ago, the order Primates was separated from colugos. Primates include both Haplorhini and Strepsirrhini, which includes lemurs and lorises. Haplorhini split 63 million years ago and includes Simiiforms and Tarsiers. Simiiforms include Catarrhini and Platyrrhini, the New World monkeys. And 35 million years ago, Catarrhini branched from Simiiforms and includes Hominoidea and Sarcopithecodea, the Old World monkeys. Now we've arrived at the superfamily Hominoidea, the apes. Hominoidea split 25 million years ago and includes Hominidae and Hylobatidae, the gibbons. Hominidae, the great apes, branched 14 million years ago and includes Homininae and Ponginae, the orangutans. 12.5 to 14 million years ago, the African apes Homininae branched from Hominidae and includes Hominini and Gorillini, the gorillas. Hominini branched from Homininae 7 million years ago and includes humans and Pan, chimpanzees and bonobos. Now, at 2 million years ago, we've reached our closest relatives in the genus Homo. Hominina includes all human species, such as Homo habilis, Homo neanderthalensis, and Australopithecus africanus. Now that just leaves us. Around 350,000 years ago, Homo sapiens split from our human ancestors and are the only species of human that are still alive today. That concludes our 4 billion year journey from the first form of life to modern humans, and that's just the beginning. We could spend a whole nother video exploring the relationship between different species of humans, and we will learn about the evolution of all kinds of different animals. For now though, be sure to subscribe so you don't miss future videos. Until next time, see ya.